Hi, everybody. As you can see, I have a friend with me. It's Dr. Cannon. You all recognize him. And I'd just like to read a short introduction, if I can get through all these big words. Dr. Cannon is an MD, PhD, and chair of the Physiology Brain Research Institute at UCLA. He is the professor in the Department of Physiology in the David Geffen School of Medicine at UCLA. He received both a BS and an MS in mechanical engineering at Washington University in St. Louis in 1980. His master's thesis explored how increased muscle stiffness and intensity of stretch reflex with mechanical loading produces an instability that may cause tremor. Upon graduation, he entered the medical scientist training program at Johns Hopkins, where he worked with Professor David Robinson to identify the locus of the brainstem neural integrator for the ocular system and demonstrated how lateral inhibition is a critical neural network feature of his premotor circuit. He completed a medical internship and neurology residency at Massachusetts Hospital, where he was chief resident in 1990. Dr. Cannon then completed a research postdoctoral fellowship in David Corey's lab, where he made a fundamental discovery of the sodium channel defect that causes susceptibility to periodic paralysis. Based on this new work on channelopathies of muscle, he started a lab in the neurology department at Harvard Medical School, where he was on the faculty until 2002, when he moved to the University of Texas Southwestern Medical Center in Dallas as chair of neurology. In 2010, he became associate dean for undergraduate medical education and developed a combined BA-MD training program with the University of Texas in Dallas. In 2015, Dr. Cannon was recruited to UCLA as the chair of physiology at the David Geffen School of Medicine. So welcome, Dr. Cannon. We're happy to see you again, and I'm going to turn it over to you.
Thank you so much, Linda. I think the audio is registering, and I'm just waiting for the screen to pop up.
Yep.
Which is coming, and I have 3 monitors active, so I'm not sure which screen you're seeing. Sorry, are you able to see my slides? I'm not sure.
Yeah, no, not yet. I see connected, go to webinar. See, yeah, you are the presenter. It's not this.
Let me try to swap which screen is showing. How about that?
There you go. That's good.
Okay.
I'll see you in a little bit.
Okay, fine. So first of all, I'd like to thank Linda for the kind invitation. I think this is now my 7th time of attending the PPA meeting, and I always learn a lot from every session. I've learned far more from coming and meeting you all and speaking with you than any textbook I've read or seminar I've attended. So I really appreciate your willingness to share your experiences. And I've been following along all morning, even with the challenges of COVID. This meeting has been running very well this year, and despite COVID and the fires in California and the fact that we had a magnitude 4.5 earthquake last night in Los Angeles, we're all still here, so that's fantastic. So as you can see on the title of my slide, I'd like to provide for you a review of some of the genetics and research updates in periodic paralysis. You've had a wonderful introduction from several speakers this morning. And I always like to start off by making sure everybody's on the same page. And whoops, that was not the right page I wanted to be on. Sorry, I think this just changed to a funny view for everybody. Let me move this over. Trying to get this controller off your screen. Pretty much unobstructed screen, I hope, for you now. So first of all, to remind everyone that in periodic paralysis, the problem resides in the muscle itself. So this is a view through a microscope of a mammalian muscle fiber, a single fiber that's been teased out in a dissection. So in a large muscle like your bicep, you would have tens of thousands of these fibers. Each one has a very special connection to the nervous system from a single motor neuron that makes a very special contact, which is how the information comes from the brain and the central nervous system to tell your muscles when to contract. And so in periodic paralysis, the problem is not with this nerve and how it's able to communicate information, or the special connection between the nerve and the muscle, the neuromuscular junction, but rather it's the problem in propagating the message from this connection to spread throughout the length of the fiber. Because muscle fibers are very, very large in comparison to this little point of contact. And so it's essential that this signal be spread rapidly and with high fidelity throughout the entire body of the muscle so that contraction is uniform and very consistent. If we take a cutaway view here of a cartoon, this is the nerve coming in. I just want to make you aware that not only does this signal need to travel along the outer surface of the fiber, but also propagate deep into the center of the fiber, which is where the machinery resides for converting this electrical signal into releasing calcium from these special stores shown in this blue here, because calcium released inside the fiber is the chemical signal to enable the initiation of contraction. So in periodic paralysis, the problem is in this communication where the electrical signal would travel over the surface of the fiber and down into the depths to enable the release of calcium and initiate contraction. So sorry. I want to also just make sure everyone's on the same page. We've heard a lot of wonderful introductions today about the types of symptoms individuals with channelopathies of skeletal muscle can experience. And we've also heard that it's all about the individual, and there can be great variability, which is essential in optimizing patient management. But I want to review for you the core features so that we can focus on, from a research perspective, what we've been trying to understand and what successes and achievements we've made in there and how that applies to optimizing management for individuals. So as you've heard, one of the manifestations of an ion channel disorder of skeletal muscle can be this persistent involuntary after contraction that fluctuates depending on the level of activity. Most individuals refer to this as being stiff after a voluntary movement that neurologists refer to as myotonia. Many of you recognize that the symptoms are often most severe with the first few movements after having been at rest for a while, and that it may resolve in a course of 30 seconds or a few minutes with modest level of repeated activity. This is the so-called warm-up phenomenon that happens in many forms of myotonia. Dr. Giraffe mentioned earlier this morning that some variants have this paradoxical response where with repeated muscular effort, the stiffness actually becomes worse. This kind is often sensitive to temperature, aggravated by cold, and this paradoxical worsening is the basis for the name paramyotonia. And oftentimes, in addition to going outside, in which it affects muscles that are out in the periphery of your body, so in the hands or in the face on a cold winter's day, that drinking chilled beverages will cause stiffness of the tongue and slurred speech to happen under those circumstances. So in myotonia, you have an abnormal excitability of the muscle fiber. So with the needle electromyogram, the physician can document in the outpatient setting the fact that all of this excessive self-sustained bursts of action potentials are occurring in the muscle. In addition to you feeling stiffness, at times it's possible for the physician to demonstrate myotonia by tapping not on a tendon for a normal stretch reflex, but to tap right on the belly of the muscle. And here you see this persistent dimpling, which should not normally be there and is a sign of this abnormal after activity that occurs in myotonia. So in the context of disorders of electrical excitability, this is obviously an enhanced excitability because the muscle continues to discharge autonomously when in fact it should have stopped and resolved. Periodic paralysis, which is the main focus of our talk this morning and of the association, is the exact opposite. So here, an individual will have recurrent, it's key that they come repeatedly, episodes of weakness. They typically have a gradual onset. We've heard some of the accounts today in the Strongbridge presentation that some people have rather abrupt onset, but in general, most of them evolve over minutes to even the course of a day. And in some individuals, the episodes are most commonly experienced with first awakening in the morning. The duration can be quite variable. As we've heard from Dr. Durant this morning, that can be a challenge to physicians unfamiliar with this condition to try to understand what the basis might be, because some individuals experience episodes that only last a few minutes, while others stretch into hours or even a day or more. It's also very clear that individuals cannot quite return to 100% strength for weeks at a time. So there can be very protracted episodes. The severity is also highly variable. It can be from relatively mild, where there's a self-perception, but others might not appreciate it, to complete immobility with loss of muscle tone. And we've heard from many this morning that a characteristic feature that's really helpful in the diagnosis in defining periodic paralysis is the fact that there are environmental factors that can bring on an attack or act as a trigger. So resting after a prolonged period of strenuous exercise is one of the most consistent triggers. We've heard in some individuals there can be changes in diet, so high carbohydrate content or salt content for people with hypokalemic periodic paralysis, or too much potassium in the diet for hyperkalemic periodic paralysis can trigger an event. Emotional stress as well as physical can be a common trigger factor, as well as alcohol ingestion. As you've heard, for the individual, it's very important to be mindful of triggers, to keep an accurate diary, because in working with your caregivers, this can be one of the most important strategies for minimizing the frequency and severity of episodes. It's also a very active episode of the research in our animal models of periodic paralysis, because if we could understand the trigger factors more completely, that would be a huge advantage to help individuals minimize the effect on quality of life from the frequency and severity of attacks. I will touch at several points in my talk on the issue of the slowly progressive permanent weakness. Clearly, this weighs very heavily on minds of people with periodic paralysis. It's what some people are understandably most concerned about. And as you'll hear through the talk, it's an area where we're really struggling to understand the basis of why this happens and be able to accurately predict for whom this is going to be an issue. So let's think about episodes of weakness in periodic paralysis, and I want to bring some clarity to you all of what is wrong with my muscle during one of these attacks. Clearly, it's a functional change. something that's happening on the time frame of seconds to minutes. And as I mentioned, in every form of periodic paralysis, whether it's hyperkalemic, hypokalemic, Anderson-Tawil syndrome, even periodic paralysis associated with thyroid disease, in every single case, there's a common mechanism. So let me remind you in cartoon form here, in the normal situation, this red trace is showing this electrical signal, which is a change in the membrane potential, that is the voltage across the muscle cell. So normally in a quiescent resting muscle, there's no change over time. And when it's time to contract, there's this brief electrical transient we call an action potential. And this is the initiating trigger, which causes the calcium release, and then an increase in force or tension in the muscle that follows very quickly thereafter. So for a patient with periodic paralysis who's in the asymptomatic period, which, as you all know, is most of the time, they have normal resting electrical activity. So I'm showing it in a unit called millivolts. These are thousandths of a volt. So normally, for all of us, when you are at rest and relaxed, in loose terms, the battery to tell your muscle when to contract is charged. It's charged at about minus 100 millivolts and sitting there resting, waiting to go. In an appropriate stimulus, here in this computer simulation, which is injection of a small amount of current, that tells the system it's time to go, and there's its all or none fast change in the voltage, which is the signal for the muscle to contract. What's happening in every individual with periodic paralysis is when you're experiencing weakness, your battery, in essence, is partially run down. That means the voltage is closer to zero. What happens is, although becoming more positive in a brief event is what triggers contraction, if you partially go there and get stuck, the system becomes resistant to responding. It's so-called refractory, so that even when the appropriate stimulus comes, here in this artificial simulation or from an input from a nerve, your muscle will not respond with the normal amplitude and very clear signal that it's time to go. And so there's a failure of this electrical signaling, and that is the essence of what's going on during periodic paralysis. So all of the research is to understand why these variants or mutations in ion channel genes cause a partial rundown of your battery, why that happens only intermittently, why that's associated with characteristic triggers, and what we can do about it. So how do we relate these various ion channels to the clinical symptoms? So as Dr. Giraffe mentioned in her talk this morning, there's a lot of overlap in the clinical symptoms and presentation in patients with channelopathies of skeletal muscle. There are two cardinal features, being either myotonia, which is this involuntary after contraction and stiffness, or periodic paralysis, which is, transient episodes of weakness from which there is recovery. And these two entities have been recognized by physicians for over 100 years. In working with patients, clinicians have identified different categories, so-called different syndromes, where some individuals have myotonia from birth, but they do not have periodic paralysis, hence the name myotonia congenita or congenital myotonia. Other individuals have periodic paralysis in which in the 1940s it was recognized those episodes are associated with either an increase in the blood potassium or a decrease in blood potassium. There's been a lot of focus on potassium levels in association with periodic paralysis. It's been very helpful to make a diagnosis, and importantly, to distinguish between these two categories of disease, because the management is completely different for these two forms of periodic paralysis, so it has very practical importance. There are other forms, as you've heard, where paralysis can come with paradoxical worsening of myotonia. And I've put a circle around these two conditions because in my view and many other neuromuscular specialists who focus on periodic paralysis and channelopathies, there's extensive overlap between the clinical features of paramyotonia congenita, hyperkalemic periodic paralysis, and normokalemic periodic paralysis. And I would really put those in the same category. And we've all seen families where different members in the same family who have exactly the same genetic basis for these symptoms can look like the features more characteristic of paramyotonia congenita or more characteristic of hyper-PP. So that can happen in the same family. And this is why I would lump those together. Hypokalemic periodic paralysis has the distinguishing feature that the blood potassium is low during the events. And importantly, it's extremely rare, I won't say never, but extremely rare for an individual with hypokalemic periodic paralysis to have myotonia. And that can be a helpful distinguishing feature for making an accurate diagnosis. And if it's a little bit ambiguous in terms of what the patient is able to report or the doctor can see on a physical exam, this is where the needle electromyogram can be very helpful to look for laboratory electrical evidence of myotonia. The Anderson-Tawil syndrome, as you've heard, is a variant of periodic paralysis that also involves cardiac rhythm disturbances, as well as changes in the formation of bones and body structure. And there's another form of periodic paralysis associated with thyroid disease. But in every one of these variations of periodic paralysis, we now have established that the common, well, these are all rare diseases, but most frequently, the underlying defect is in one of these little molecular machines, a so-called ion channel, that's responsible for regulating the flow of sodium, potassium, chloride, or calcium in and out of the muscle fiber. And as you can see, the different clinically distinguished entities are associated with problems of different ion channels. So chloride channels for congenital myotonia. Sodium channels, is very interesting, can cause a wide variety of different clinical phenotypes. And this is an area that our laboratory has been working on for this year, makes 30 years we've been studying these, whereas the calcium channel mutations have all been for hypo-PP, et cetera. You also should be aware that in the genome of humans and animals that there is not just one type of sodium channel or one potassium channel, but there are many, many different variants. So each of those variants is encoded by a separate gene. For example, in humans, there are 10 different sodium channel genes. It's been shown that 9 of those form functional channels. And the special one called the NAV 1.4 gene is expressed in skeletal muscle, for example. This is very relevant because this particular type of sodium channel, the one that is mutated in periodic paralysis, is only expressed at significant levels in skeletal muscle. So it is not present to significant levels in other electrically excitable tissues like nerve, brain, or heart. So that's why typically individuals with defects in these channels do not experience arrhythmia or seizures or peripheral neuropathy. This is the name that researchers give to the particular channel, which I apology for all of this jargon and nomenclature, but it's a different naming system for the gene. So these abbreviations down below are what you see in the report back from genetic testing. So it's the SCN4A gene for the sodium channel that's expressed in skeletal muscle. Or a calcium channel gene, so that's calcium channel type 1S for the calcium channel and hypokalemic periodic paralysis. Now, interestingly, what makes sense scientifically is there's one exception here, which is that these particular variations of potassium channel genes, they are so-called inward rectifier genes, that's what the IR means. These are expressed in considerable levels in the heart and in bone, as well as skeletal muscle. So this is why the arrhythmia and anomalies in bony features and so height, shape of fingers and face and things like that can be affected in Anderson-de-Will syndrome as opposed to these other channelopathies where skeletal muscle is selectively affected. So I'm going to now spend the rest of our time together telling you what we've learned about the basis for which these variations in these channels, because of these gene variations that you've inherited, why does that cause periodic paralysis? Because, you know, we strongly believe that understanding why is the first step in taking a rational approach to optimizing interventions to modify the burden of disease, help improve your quality of life, and maybe even someday be able to offer a cure. Now, I'm going to tell the story primarily in cartoon form. If there are any who are watching today that are really interested in some of the hardcore science behind this, 2 years ago, I gave a talk at an international channelopathy meeting in Chicago. Strongbridge was kind enough to have a professional recording of that, and it's available as a Vimeo online recording. Here's the URL for the website. And what's also some of you might find interesting in this presentation is I used that opportunity to really highlight the enormous contributions from Professor Frank Lehman-Horn, who many of you know came to many sessions of the Periodic Paralysis Association and was really a pioneer in understanding periodic paralysis, but unfortunately passed away a few years ago. So let's get on with what's wrong with these channels. Why do I get episodes of weakness? Why is my battery susceptible to partial rundown? So I'm going to start with and focus a lot today on the sodium channel. So it turns out that for these variations of the clinical presentation of hyper-PP, paramyotonia congenita, as well as normal kalemic periodic paralysis, for these individuals who have sodium channel-based myotonia but no paralysis, for all of them, they share a common problem in the way the variants or mutations affect the behavior of the channel. So here in cartoon view, what I'm illustrating for you is that what a channel does is it helps these ions, these little dissolved salts. So for example, sodium, these little balls are to represent sodium ions in solution to move from outside the muscle fiber to inside. This orange line here represents the barrier between the inside of a muscle fiber and the outside. Biologists call this the membrane. I'll refer to it as the cell membrane. And so these channels literally form a conduit or a pore between the inside, between the outside and the inside. And in order for this delicately tuned system to work properly, it's essential not only for this access pathway to be formed to help the signaling occur in your muscle, but it has to be open and at the appropriate time. So there's conceptually a gate shown here that's occluding this pathway that might be open or closed. And it needs to be open only very briefly at the appropriate time to allow this electrical signaling to normally occur. So for all of these conditions, the problem is the mutation occurs in the part of the channel that's regulating the operation of this gate. And the gate is open inappropriately more than it should be. So it's a gain of function, more sodium comes in, and that's the basis for all of these channelopathies caused by sodium channel defects. Interestingly, for hypokalemic periodic paralysis, and what was a real challenge in the field for over a decade of having identified these mutations, is that the first mutations were identified in the calcium channel. Bernard Fontaine's group in Paris, working with Frank Lehman-Horn in Germany, mapped this to chromosome 1. Louis Ptacek in Utah, and then San Francisco was the first to find a mutation, and then Mike Hanna's group in London has found many more, so it's been an international collaboration. Initially, it was thought there was this nice distinction. Hyperkalemic periodic paralysis was going to be a sodium channel disorder, and hypokalemic periodic paralysis was viewed to be as different because it's a calcium channel defect. Well, about 8 years into this story, it was discovered that many families who clearly had hypokalemic periodic paralysis did not have a calcium channel mutation. And Angelica Hahn in Canada had a large family with this scenario, and Dennis Bowman, who's a molecular geneticist, helped her look at these families and found that in fact they had a sodium channel mutation. And initially there was a lot of resistance. This is now about 1999, that many experts in the field thought, well, this was just some anomaly, a single family. This was not going to be the case. But it has now turned out that many investigators went back to the samples in their freezers for families who clearly had hypo-PP characteristics, but no calcium channel mutation, and they had sodium channel mutations. So now we know that about 60% of families where there's a clear history of inheritability with a hypo-PP phenotype will have a mutation in the calcium channel, and about 15 or 20% in the sodium channel, and there's still another 20% for whom we don't know. So this was very satisfying in terms of helping patients and understanding of the genetics of the disease, but it was a real mystery for physicians and scientists who study the way excitability is regulated in a muscle fiber, because sodium channels and calcium channels do very different things. They have different jobs that they perform in the muscle. The sodium channel is responsible for that brief electrical transient, the action potential that we talked about, whereas in skeletal muscle, the calcium channel is a sensor of a voltage change. It doesn't cause it to happen, it senses it and triggers when the calcium should be released. So for two types of channels with very different jobs, it was hard to understand how mutations in either one of these could cause the same clinical scenario, because there must be some overlap. And finally, the insight came in about 2007. Our lab and another lab discovered that in this situation, both channels, whether it's a sodium channel or a calcium channel, becomes leaky. So it's not a problem with the regulation of how sodium or calcium comes through the normal conducting pore, the normal channel that lets these ions come in and out of the cell and regulate excitability. But instead, there's a little sidebar here, another part of the channel complex shown with these little pluses here, because part of the channel is sensitive to the voltage across the cell. It's the so-called voltage sensor. And in hypo-PP, that's where the mutations reside. They're clustered there, which I'll show you in the next slide. But what it does is it causes a leak through the voltage sensor, not in the gates that are regulating the inflow and outflow of ions through the channel. So it was a completely different class of functional defect. It explains why this was a different category of clinical scenario with different symptoms, but it also explained why this could occur with either a sodium or a calcium channel, because this voltage sensing mechanism is conserved for both channels. It's evolutionarily conserved. And so the molecular defect in either one of these could cause a similar functional defect. So here's a different view. In the gray bar, here's our membrane again. This is a ribbon cartoon of the sodium channel. This is the calcium channel. You can see these cartoons look pretty similar, and that's to reflect the fact that in real life, there's a great deal of similarity in these proteins that are coded by two different genes. Each one of them has about 2,000 amino acids, and the genetic mutation is what's changing one individual amino acid. What I want to remind you of in this slide is that over the years we've identified in this diagram here that there are certain parts, certain places along this string of amino acids where this part of the channel controls its voltage sensitivity for either one. And what I want to point out is that of the 11 mutations that have been identified in families with hypokalemic periodic paralysis in the sodium channel gene, all 11 of those occur in these voltage sensing domains at a particular type of amino acid that's the arginine. There are 20 different amino acids that can be encoded by a particular gene at one location. We use the abbreviation R, so they're all R's here, but look at this. All 11 of them are a missense mutation at an arginine. And in the calcium channel, 8 of the 9 described in literature are also missense mutations at arginines, all in the voltage sensor. There's one exception. It's been proven it causes the same functional defect. These all cause the leaks. Okay, so why am I going into this minutia? It's because this can be very helpful in a genetic screen if you were looking for the basis of hypokalemic periodic paralysis. So while this will not explain the inheritance of every case of hypo-PP, it explains the overwhelming majority. And so you should, as a physician and molecular geneticist, you should expect to see a variant in either one of these genes that affects an arginine in the voltage sensor domain. If that's not the case, you really have to wonder whether this is a benign variant of unknown significance and not the basis of hypo-PP in this family. So this is an example where knowing a little bit of detail about the science that goes behind this can help in a very practical way of interpreting a genetic test. What I'd now like to do is explain to you why this unusual leak that's created by these mutations causes susceptibility to hypo-PP and how we can use that information to strategically design interventions. So there are many different approaches to this. What we have done is genetically engineer the mouse. So there are no natural, you saw a video this morning from Dr. Girath of the infamous quarter horse, where that was a spontaneous mutation that causes hyperkalemic periodic paralysis. A wonderful opportunity to learn more about periodic paralysis, but it's a little cumbersome, as you could imagine, to have horses in the laboratory and raise them, although the University of Pennsylvania has done that in their veterinary school. And I've had the opportunity to actually see those horses. It's quite amazing. But a spontaneous mutation that causes hypo-PP has not arisen in mice. So we use genetic engineering to create the sodium channel mutant mouse. And as the advantage of this is we can study the mouse muscle under very controlled circumstances. So we remove the calf muscle of the hind limb of the mouse. It's in this little jar here in solution. A nice warm bathing solution. Here in pink, you can see the calf muscle of the mouse that we can now electrically stimulate and control the environment and measure the change in force that occurs in this mouse muscle. So here's a typical response. This is time and force. These are individual trials for mice that have a different genetic background. Just like humans, mice have 2 copies of every gene, including the sodium channel. So the plus indicates the normal mouse. So this is a normal mouse with 2 normal copies of the sodium channel. And if we apply a series of electrical pulses for about a half a second, there's a very rapid and robust increase in force in this mouse muscle. And if we challenge this mouse with, instead of being at about 4.7 milliequivalents of potassium in the bathing solution, which is what you have in your normal body, but decrease it to 2 millimolar, the normal mouse tolerates that pretty well. There's only a modest change in force. But if we take a sample from a mouse that has 1 mutant copy and 1 normal copy, so this would simulate what happens with patients who've inherited this mutation. Now, when we give the 2 millimolar potassium challenge, there's a clear and dramatic reduction in force. And what we're able to do in mice, which is one of the advantages of this laboratory model, is we can interbreed the carriers and have a so-called purebred mouse that has 2 copies of the channel mutation. These mice appear normal. They're able to feed and reproduce and run around the cage, but they're much more susceptible to hypo-PP. So in this mouse, if we expose it, here's the baseline force is less than normal in blue. And if we give it a 2 millimolar potassium challenge, you see the force drops all the way to essentially 0. And then if we return to normal 4.7 millimolar potassium, it recovers. So this shares all the features of susceptibility to low potassium-induced attacks, and they are reversible. So this very clearly shows that it is indeed possible to have a single genetic variant in the sodium channel and have a hypo-PP variant, just like we've seen in patients. I want to show you similar results from our model of a calcium channel mouse. Instead of showing the raw force traces, I'll now talk about the maximal level of the force. So how much force relative to normal is able to be generated. So we've made a mutation of the R528H variation in our mouse. This is the most common cause of hypo-PP in humans. This particular missense mutation. And sorry, I didn't change the label on the mouse here. We've actually made the R528H in the calcium channel. And now what I'm showing you is similar to these results. We're showing the relative amplitude of the force, starting out being 100%. And we're doing this test of measuring the force every 2 minutes for an hour, for 60 minutes. And over the first 10 minutes, the force is very reproducible and very stable. And if we then challenge this muscle with 2 millimolar potassium, for the normal mice, there's a little bit of a decline, but the force is relatively well preserved. Whereas for these male mice in dark, there's a 60% loss of force. As long as the potassium is low, the force stays low for 30 minutes, and then when we return to normal potassium, here's the recovery. So again, showing a transient episode of weakness, all the characteristic features of hypo-PP. I want to point out to you that interestingly, in the female mice, in a 2 millimolar potassium challenge, they can become weak as well, a little bit more slowly for the mice that have a double copy of the mutation, but the mice with a single copy are more like the wild type. So this is not a low enough potassium challenge for them to have a trigger. And this occurs in human patients as well. As many of you know, in particular with the calcium channel mutation, the R528H, Tara Links has published on this in the 90s, large family in the Netherlands, multiple affected generations, where many women do not have episodic attacks of weakness, although unfortunately they can get the permanent progressive weakness. And so, you know, this is an opportunity to learn more about this gender specific basis for susceptibility to weakness, and really demonstrates the amazing high fidelity of these animal models. I want to stress to you that we recognize mice are not humans, that these are approximations of human disease. But there are a lot of mouse models of disease, of cancer, of neurogenetic diseases, of autoimmune diseases. But these channelopathies of periodic paralysis are some of the best high fidelity models of human disease that we have. And here's another example of reproducing those characteristics of the human disorder. So we took this muscle and challenged it not only with 2 millimolar potassium. So here we're changing the potassium, that we're challenging this mouse model. We do it outside the body in a dish, because of course in the body there are a lot of mechanisms where, just like in a human, the mouse's body is going to attempt to regulate the potassium level, and we can't as easily control it in an experimental manipulation. That's why we take the muscle out of the body and we can vary the potassium. And what I want to show you is this is essentially a dose response curve. So indeed, if we increase the potassium, there's no difference between the maximum level of force of these mutant mice and the wild type. The problem is in low potassium, and you can see a dose response curve. The lower the potassium gets, the worse the problem is. And it gives us some sense for when this muscle begins to be in trouble. You could never get this data from humans. You need to do this on a muscle fiber itself. You can't do this from a skin biopsy, growing fibroblasts, even doing fancy, induced pluripotential stem cell research. You just can't do this. You need whole muscle. Frank Lehman's Horns group did pioneering studies on patient biopsy material. We really appreciate the participation of those patients because you're talking about an open biopsy to have a large muscle sample. But that preparation is viable for about 12 hours. So only limited studies available on measuring force, which is an example why these mouse models have been so important to establish this information. So now what we've shown is we've reproduced the model. We have found out that it's leaky channels, but how do you connect the leak to the risk of being susceptible to a loss of force in response to potassium? And I'll show you the story gets even a little bit more involved because it's going to involve chloride as well. So I don't want to overwhelm people, but I just want to point out some of the important players that come into consideration of understanding why this leak puts you at risk of having your battery partially run down and have periodic paralysis in the setting of low potassium. So this cartoon of a cylinder represents the muscle fiber, and there are many different ion channels. Here I'm showing 2 different potassium channels and a chloride channel. There are pumps that use the metabolic energy of ATP by burning glucose to establish a concentration difference of the salts between the inside and the outside of the fiber, and other transporters that bring ions in and out. And all of these, in an orchestrated way, regulate the excitability of your muscle fiber. And by understanding how each of these players contribute, we can use computer simulation to ask, how well is your battery charged? What is the voltage across this muscle as a function of the external potassium? This is your blood potassium level, which normally is between 3.5 and 5. So normally you're in this range, and in the normal situation, the model cell says in this range of potassium, you go up to this black line, your battery would be pretty well charged, very well charged, and you're all set to go and would enjoy normal excitability and strength. What happens is as the potassium in the blood and outside the muscle fibers gets lower and lower, which can fluctuate for changes in diet and exercise, diarrhea, sweating, a lot of things can change the potassium level. It bounces around. For most of us, it's not a problem. In your body, it would be extraordinarily rare for potassium to get much below 2.5. You begin to have a lot of problems with rhythm disturbances of the heart and other electrically excitable tissues. But what I will say, and what helped us understand it, is that even if you study normal muscle from patients without periodic paralysis or mice or other organisms, is that if the potassium becomes extraordinarily low, lower than could ever be achieved in the body and be compatible with life, but if you make it low enough, the battery partially runs down. So instead of the battery getting stronger and stronger and stronger and better charged, this paradoxical shift happens. And while that sounds like a laboratory esoteric curiosity, this was really the insight that helped us understand what happens in hypo-PP. Why does your battery run down in low potassium? Because if you take this scenario and now introduce the leaky channels that cause hypo-PP, so there's an inappropriate inflow of a little bit of sodium because of a mutation either in the sodium or the potassium channel, then the impact that that has on this computational stimulation of excitability of muscle is it shifts this relationship to your right. What that means is this transition as your potassium is getting lower and lower outside the muscle. The normal random variability here in the potassium level, now when you're down at the low normal level, there can be a problem where the battery partially runs down, your muscle fiber is now refractory, it's inexcitable. This will cause periodic paralysis. So it's this shift caused by this mild leak is why you have hypo-PP. So that's important because if we could modify the behavior of either the mutant channels or perhaps other contributors to electrically excitability of muscle, there's the chance we could shift this back and get you out of trouble and thereby improve the management of episodes of weakness in periodic paralysis. There's one final point I want to make this, and then we'll get back to reality a little bit and away from these theoretical diagrams. But I want to point out the variation in potassium and the symptoms you experience. Now, it is, again, a very important feature of these diseases that the susceptibility and the risk of having an episode of hypo-PP is dependent upon the potassium. And when potassium gets lower, you increase the risk of an episode of weakness. But it's important to realize, and as shown in this diagram, that if you're in a lowish level of potassium, like around 3.2, so you go up a vertical line here, my potassium is 3.2, and I have a mutation, so the dotted curve is the relevant analysis here. You could be sitting here, which is well-charged, good membrane potential, and you will be asymptomatic. But with this very same lowish potassium level, you could be up here with your battery sufficiently run down and symptomatic and inexcitable. The point is, it's not just the potassium level, it's also what state you are. And that's why, as you've all observed, there's a biologic complexity of interpreting your potassium level and am I going to be weak or not? Because some people with hypo-PP, this curve for the individual could be stretched over this way even further. Some people with relatively normal potassium can have an intermittent attack, and I'll show you that in the mouse model. On the other hand, if potassium is low enough, sure, everyone will get an attack. But there are these interesting overlap regions where you might or might not have an attack. And we know that clinically from what patients tell us. We see that computationally in the model. It's right here in the fold in this diagram. And we see it in the mouse models. So that's a really robust part of this condition. And you need to be aware of that because you should not overinterpret a particular potassium level. It's valuable and in keeping your diary to know what's my level and where do I get in trouble, but don't overinterpret it. And physicians need to be aware of this because if they see a patient in the emergency room, there can be a difference in time between when the attack occurred, whether the person took potassium, where they are in the recovery, and can it be really difficult to use the potassium level in establishing a diagnosis. And this illustrates some of the complexities there. So what we'd like to know is when is an individual going to be on this part of the curve and be asymptomatic and have normal strength, and when are you going to be up here? And again, I don't want to be overly detailed about this, but we've learned that the amount of chloride you have inside your muscle is effectively the switch that biases the system to whether you're going to be down here and asymptomatic or up here. And if the chloride content in the muscle is low, you'll be in a favorable situation, and if it's high, that'll be unfavorable. So I want to be clear that there are many mechanisms, this channel and this co-transporter, that in very sophisticated ways regulate how much chloride is inside the muscle compared to outside. It's much, much lower inside than outside. We'll talk about some medications and things you can do to bias that balance and improve your opportunity for being asymptomatic. But please don't misunderstand this and think by varying the chloride in your diet or what you consume that you can affect this balance. There are other ways we need to approach that. So in theory, the idea was if we could keep the chloride low inside the muscle, you would avoid this and things should be better. The main mechanism by which chloride becomes elevated inside muscle and other cells in the body is this transporter. This so-called sodium-potassium 2-chloride transporter is in every cell of the body, and it responds to changes in the number of things that are dissolved in the cell, whether you're hydrated or dehydrated. I'll get to that in a moment. But this transporter is very strongly inhibited by this potent diuretic called bumetanide or Bumex. So the hypothesis in just understanding the science behind periodic paralysis was that if you could inhibit this transporter with this drug, that might be beneficial for hypokalemic periodic paralysis. So we tested that in our animal model. So here's the sodium channel mutant model of hypo-PP. We're studying the muscle in the dish from this animal. Here I'm showing the raw data, the electrical stimulation for half a second. Here's the normal maximal development of force in normal potassium. We take this same muscle and lower the potassium to 2 millimolar, which for a patient with hypo-PP would certainly get you in trouble. And the force is lower in this animal model. Even staying in 2 millimolar potassium, but now adding 1 millimolar of bumetanide on top of that, there's a dramatic recovery of increase in force. So here's a rescue of weakness within a few minutes by applying a realistic level of bumetanide that you could achieve in the body. We performed this study on the calcium channel model of hypo-PP as well. Again, this is the most common inherited form of periodic paralysis. The control amount of force. If you challenge with low potassium, here's the attack of reduced force. You stay in low potassium and add bumetanide. There was a partial recovery, in most cases not complete. Just to show that the muscle is still healthy, if you go back to normal potassium, there's recovery. I will say that if you do this in a preventive manner, so administer the potassium, the bumetanide before the potassium channel, you can be completely protected. So there was a lot of enthusiasm for this as an opportunity for management of periodic paralysis. I apologize. I'm going to, I know I'm 55 minutes into the talk, and I'm going to look for messages from the organizer. Linda was kind enough to put my talk in the end. She knows I tend to go long. I have some more content I'd like to have. I'm not going to shortchange the questions, but I want to make a few other important points about this hypo-PP story and this transporter. First, I'd like to
feel free, Dr. Cannon.
Okay, thank you. And just pop in. I'll say that in trying to manipulate the multiple monitors I have on my computer, I cannot live see the comments from the moderators, so please just break in with audio and I'll adjust. So another really exciting area just in the last 2 years that we've been able to explore with these mouse models is the connection between exercise and episodes of weakness. Because while this is clearly recognized by patients and physicians as one of the most consistent triggers, we had only guesses. There was no clear understanding of how to connect those dots. And so it was hard for us to develop strategies to help protect people before they wanted to exercise and again, improve lifestyle. So this actually started back with Frank Lehman Horne studies on biopsy samples from humans in the 1970s and 80s, as well as some insights on the way that acetazolamide and dichlorphenamide might be benefiting. And it focuses on the fact that under conditions of really vigorous and extreme exercise, in part of the metabolic process of burning fuel, burning glucose and things like that by your muscles, a byproduct is acid. And the muscles themselves and your blood develops higher levels of acid during exercise. A curious thing about the weakness in periodic paralysis is it comes after exercise, not during exercise. So while we all recognize that exercise, if you will, is the trigger, the curious thing from a mechanism is that somehow exercise is protecting you. And so Professor Lehman Horne in the 70s and 80s tested this with human muscle fiber strips in the lab and showed that in hypo-PP, if you lower the potassium and the muscle becomes weak, in the laboratory, if you increase the acid in that bath, you can cause that muscle to recover. So he had this insight. So we followed it up 30 years later when we have the genetic model of the mouse. And so what we're showing here is, again, our experiment of the mouse outside, the muscle outside the mouse body. We're monitoring the peak force, measuring the force every 2 minutes. This is an hour-and-a-half experiment. Every dot is a separate measurement. And what we varied, we didn't actually exercise this muscle with repeated contractions, but in the bath, we changed the amount of acid. So you may remember from your high school chemistry and biology, pH refers to the amount of acid. Lower pH means more acidic. So what we did is for 30 minutes, we made this bathing solution more acidic for either a normal mouse muscle or our 2 models of hypo-PP or our model for hyper-PP. All the different variations of muscle fiber tolerate this acidosis just fine. You see the muscle force stayed relatively preserved. But when we quickly went back to a normal pH, so this is what would happen after exercise when you rest and the acidosis recovers. What happened in the 2 models for hypo-PP is there was a dramatic decrease in muscle force. It didn't happen for normal muscle, did not happen for hyper-PP. Now, I will acknowledge patients with hyper-PP have exercise-associated weakness. So a different mechanism is happening. Something else is going on. We need to do more science. But for hypo-PP, what was curious is regardless of whether it's a calcium channel mutation or a sodium channel mutation, both of which give leaky channels, you're at risk of this, so-called after exercise loss of force. And I will tell you from studying hundreds of muscle fibers in the laboratory from our mouse models, this recovery from acidosis is a much more consistent trigger for a loss of force in our mouse models than the potassium challenge. And potassium has that fuzziness to it, and I think many patients and you all who are listening out there, drop me an email and let me know, because I learned from you. Patients tell me that really the exercise phenomenon is one of their most consistent triggers, even more so than potassium. So I'd be interested in knowing about that because that's what we see in our animal models. So what we'd like to know is the same mechanism we saw for the low potassium challenge. We showed you that blocking this transporter with bumetanide was very effective for the low potassium triggered loss of force. What about these so-called exercise-induced loss of force? So we looked at it 2 different ways. Sorry, I got ahead of myself. One is how quickly you recover from exercise. So you all have told me, so I took my lead from your story, that if after exercise, if you recline on the couch, and we heard this from some stories about patients this morning, but that's a high risk behavior to trigger an attack. But if you slow down and warm down your exercise, or if you start to feel the onset of an attack, you resume exercising, you can abort an ongoing attack. So we tried to confirm that phenomenon in our animal model by not having an abrupt recovery of the acid-base balance, but a very slow recovery. And that's what I'm showing here. So these lines are actually measuring the amount of acid in the bathing solution. So in both cases, for our hypo-PP muscle, we make it more acidic. This is the exercise period. In one muscle, this was from one leg, we rapidly correct the acid, and in the other leg, the same animal, we slowly correct the acid. And look at the difference. The rapid correction is a much greater susceptibility to loss in force than slow. So again, the fidelity of these animal models for helping us in the laboratory to in detail investigate what you have told us and help us understand why this difference is beneficial. What I will say is that immediately this tells me that the stories that you relate to us have a strong mechanistic scientific basis, and I really put a lot of credibility into that warm down or re-exercising, and you should be aware of that. Your doctors who take the time to listen to you have been making this advice for years, and I would just encourage everyone to do it because we can see it's a very robust phenomenon. So go back in sequence. We also tested bumetanide for the exercise situation. This is my abbreviation for bumetanide. So if you don't administer bumetanide, this simulated exercise causes a dramatic loss of force. But in the paired muscle from the other leg, if you pre-treat with bumetanide, you can completely prevent the loss of force. So not only potassium-induced triggers, but also bumetanide. There's another interesting consideration that relates to the video that was shared in the Strongbridge vignette. You heard about someone for whom dehydration is a trigger for their attacks. And that's the first time I heard that story, actually, but you all have told us that if you eat a lot of potato chips or pretzels or have a high, not potassium salt, but sodium salt ingestion, that can increase your risk of having an episode of hypo-PP. And that's very interesting because this co-transporter here, its normal job is to regulate the balance of salt between the inside and the outside of cells, because if you have a lot of dissolved salt, so-called hypertonic situation, or if you're dehydrated, that'll cause your cells to shrink, or in the other direction to swell. And to regulate and compensate for shrinking and swelling, that's the main job of this transporter that we're inhibiting with this drug. But there's another way to modulate this, because that is to regulate its level of activity. If you're well hydrated, drink a lot, don't eat a lot of potato chips, that will naturally decrease the activity of this transporter. So that's similar to taking this drug. So the prediction is, if you stay well hydrated, you'll slow down this activity, you'll have less chloride inside your muscle, that could be beneficial. Conversely, if you're dehydrated because you haven't been keeping up with fluid loss, or you eat a lot of potato chips, that's going to cause this transporter to be very active. It's going to bring chloride into the muscle, and you could increase your risk of having an attack. So we studied that in the dish. So we can just control the amount of so-called hydration or dehydration. And what's new, we published some of this earlier last year, but in what's unpublished is that we can change the amount of hydration, which is what your physicians call osmolality. It's something they can measure in your blood. It's normally has a value of about 290 or 310. And in our paper, we had huge changes in the amount of hydration because scientifically, we just wanted to explore a proof of principle. But we've gone back and revisited that, looking at more modest changes in the amount of hydration. So what we've done is our mock exercise experiment in a hypo-PT muscle. Here's the normal amount of force. We have simulated exercise in normal level of hydration. That's a risk for triggering a loss of force. If we take the paired muscle from the other leg of the same animal, but before the simulated exercise, we just dilute the bath solution a little bit by 7%. The muscle tolerates that just fine. That dilution doesn't cause any change in force. But now the recovery after exercise is completely protected. So reasonable amount of hydrations, of hydrating yourself, has a real scientifically verified opportunity to improve protection from having an exercise-induced attack. Conversely, if we induce simulated dehydration, this is a change from 290 to 310 for the physicians in the audience, a modest change in dehydration and increased osmolality. Even before the simulated exercise, the muscle that experiences this begins to have a loss of force. So dehydration is bad. Interestingly, when you induce the acidosis, the muscle recovers a little bit. This is the phenomenon of something that goes on during exercise is protective. You don't get an attack during exercise. This is probably also how carbonic anhydrase inhibitors are working, because they cause your blood to become a little bit more acidic, and that's protective. But now when the simulated exercise is abruptly corrected, without protection, of course, you have an attack of weakness. But in the dehydrated case, it's even greater loss of weakness and even prolonged period of time. So again, very robust, consistent evidence that dehydration is clearly bad, hydration is better for you. The other thing I'll point out, and then I think I hear Linda coming on the audio, is that all of these phenomena you're seeing, a dramatic loss of force in a model of hypo-PP, was all induced when the potassium was normal at 4.7. So I want to remind you that it's a combination of features and susceptibilities that comes into play with whether or not the excitability is going to be compromised and the muscle is going to become weak. So the level of potassium is one thing, but the balance of the chloride in the muscle and hydration is another. So we have to be open-minded about considering multiple factors that come into play for regulating excitability. So these were some word slides that I'll put out here, which just it's reiterating what I've already said about what we've discovered in the laboratory, how that helps with diagnostics, how that helps with recommendations for lifestyle, and what that implies for symptomatic management. And the slides will be on the website, and you can see that. Linda, I have about 5 minutes without data, but to talk a little bit about future in diagnostics and gene therapy for periodic paralysis. I can take 5 minutes, or we can switch to questions, whichever.
Go for it. Go for it.
Okay. So I'd like to, in the last couple of minutes, be forward-looking for the future horizons on advances in periodic paralysis. So to show you how recent this is, this paper was published just about a month ago. And this was a collaborative study on a lot of individuals who are studying the bioinformatics of inherited diseases, many types. There was a focus on diseases caused by mutations of ion channels, but not just for periodic paralysis, also for epilepsy and for arrhythmias of the heart. The epilepsy situation has been particularly challenging because there are hundreds of mutations that have been identified, way more than could be studied individually in the laboratory. And so you need some overriding principles in bioinformatics to begin to make sense of this. So we face this same challenge in periodic paralysis. I will say that in the sodium channel story, if you combine hypo and hyperkalemic periodic paralysis, to my list and the publicly available lists, there are now maybe 70 or 80 mutations that we're pretty confident cause susceptibility to periodic paralysis. For the calcium channel, there are fewer, more on the number of about 15 published and another 5 that are on the verge of being published. But any of you who've gotten laboratory results back from Invitae or Fullerton Labs or others, these are high quality laboratories. The accuracy of looking for changes in the genetic code is robust. It's not detecting the change, it's interpreting the change that's challenging. And so you get back this dreaded report VUS, which stands for Variant of Unknown Significance, because the science is still at a place where, although we are sure there's a variation in your genetic code, we don't know what the impact of that change is yet on the way that channel will function and whether that actually increases your risk of periodic paralysis. So what this paper did was, again, a bioinformatics, sophisticated machine learning approach at cataloging all the genetic tests that have been done, all of the science that's been done in studying those channels and trying to put this together. And what I'll show you is one example. So here's our friend that I showed you before, which is a cartoon of the sodium channel. The calcium channel would look very similar. What these investigators are sharing with you, here's the membrane separating the outside from the inside. And like all channels, this protein, what the gene is coding for, with 2,000 amino acids, folds in and out of the membrane. That's how it forms the ion conducting channel. What they've color coded here is from all of this literature, what location on the channel have there been variants associated with pathologic changes that affected the way the channel behaves and caused symptoms in patients versus variants that did not either change the way the channel behaved or consistently show symptoms in patients. And so this kind of mapping is going to help us and help Invitae. They use very sophisticated algorithms. I've spoken with them. I'm very impressed with the service they provide. But this is going to be another tool to help interpret this genetics and hopefully reduce the number of variants of unknown significance. So I just wanted to make you aware that there's additional developments there. Invitae is on board with this. All of these entities are consulting the scientists who study these channels, and so there's reason to be optimistic that the interpretation is going to improve. So that's great. In terms of therapeutics, you saw in my examples before working in the mice, we're really getting a better resolution at being able to provide you recommendations for lifestyle changes and drugs that can modulate susceptibility. And I'll tell you, there's more exciting stuff on the way that we're working on, hopefully in the not too distant future. But I really want to get to this question because it came in to some of them advanced questions that came in to this year's webinar and meeting. What about this slowly progressive weakness, permanent weakness? It weighs heavily on everyone's mind. We've heard about the impact it can be on mobility. It's vitally important, and it's frustrating because the amount of information that's available is much less. Even today, the precise mechanism is not well understood, not for lack of trying. Lots of work has been done both on human biopsies, looking at trying to understand how channels affect it, examining the pathology of these muscles, looking at metabolic changes. And we're still looking for it. I will say that in the mouse models, people will say, you've got the models, why won't you help us? Why won't you look at that? And the challenge is, so at any one time, we have about 400 mice in our laboratory with periodic paralysis. And we've had these mice for over 10 years. And in all that time, in all those mice, I've only seen about 2 or 3 mice that developed permanent weakness and were dragging their hind legs. And we studied those very carefully, but unfortunately it was unrevealing. So it's a very rare event in the animal models. So that's making it hard. The other thing is to have, we all know that one of the more consistent features of this in patients is the risk increases with advancing age. You don't have to necessarily be 50 or 60 years old. It can come earlier, and physicians need to be aware of that, but everybody recognizes the risk increases with age. Well, the equivalent of a 40 to 60-year-old human is about, a 14 to 16 or 18-month mouse. And when you're trying to get funding and sponsor students and do studies on aging, that's a huge challenge in doing animal models of disease for aging. So there are very real challenges, and I just wanted to make you aware of them, and it's not for a lack of trying or a bias or undervaluing the importance of studying permanent weakness. I want to speak to what is out there because it's important for you to think about managing your symptoms and optimizing the way you deal with periodic paralysis to try to minimize the development of permanent weakness. Physicians who've seen lots of patients, more than myself, and have looked at this for years are convinced, we don't have double-blind trials because it would be unethical to do it, but trying to look at natural history and historical data, that it's not good for your muscle to have these recurrent, repeated attacks, and that repeated severe episodes is not a good thing and is probably increasing your risk of permanent muscle weakness. On the other hand, it's very clear, and again, some of the best data comes from Tara Links and her colleagues in the Netherlands. Beautiful studies, multi-generational family with genetically documented R528H, for whom many, many individuals, especially women, developed significant proximal muscle weakness in advancing age, who never had a recognized attack of acute paralysis. So there's clearly a dissociation that occurs. So I would not say episodic attacks is a prerequisite, but it still makes sense to do the best you can to minimize the severity and frequency of attacks to decrease the likelihood of this happening. So, and again, we don't have an established animal model. And so really the opportunity for permanent weakness is looking more towards the cure. And again, I apologize for my time, but in the next 3 or 4 minutes, I want to tell you where we are on gene editing technologies. So here the idea would be to actually, in patients, engineer the change, the missense mutation that you have that's putting you at risk. Because this is like the home run swinging for the fences. Even if we don't understand the mechanism of putting you at risk for permanent muscle weakness, it makes logical sense that if we could correct the genetic defect, that should reduce the risk and hopefully stop the progression down the road to permanent muscle weakness. So let me remind you of where we are. And a lot of you have seen on the news for the past several years, this CRISPR-Cas, which is a molecular tool to edit DNA. It was discovered in bacteria. Actually, people studying cultures to make yogurt in France was actually the discovery. So put a plug there for basic science of genetic mechanisms. And the amazing thing is these enzymes that cut DNA in a very special way work in mammalian cells and can work in human cells. So the big player in this is the Cas, which is shown in gray here, which is a pair of molecular scissors that can cut DNA. So the whole technology here is to be able to cut your DNA to deal with a mutation and perhaps do some useful editing. Of course, you have to cut in a very precise, special place. And that's from this other piece, which is a little strip of genetic material. Instead of DNA, it's RNA. So it's not deoxynucleotide ribonucleic acid, but it's ribonucleotide acid, RNA, tongue twister. And it's this RNA that helps target the molecular scissors to tell where to cut. Now, it's easier to make cuts and destroy a little site on DNA than to make repairs. That is, in periodic paralysis, you are actually in a favorable situation for using that type of gene editing. Because as you've heard, many cases of periodic paralysis, the reason why it's genetically inherited, you only need one copy to become symptomatic, is because the mutant copy is a bad actor. So it's actively doing something deleterious. So if you could go in and disrupt the mutant copy, you'd get rid of the bad actor. That's part one. But part two is we already know from both human genetics and animal models is that you can get by perfectly well with one good copy. So it's the one bad player that's messing things up. If you reduce the number of copies to one, but that one copy is good, you will be fine. So this is well established in animal models. We actually deleted one copy of the sodium channel gene in mice, and they're fine, and they do not have periodic paralysis. And the group in London, the University College London, Francesco Mutoni, working with Mike Hanna and his group, studying a different disease, congenital myopathy, stumbled across recessive mutations in the sodium channel and showed that in patients, if you're missing one copy, you're going to do okay. You're at risk for a mild congenital myasthenic myopathy syndrome, but you're not going to have periodic paralysis. So that's robust evidence that we could, in theory, cut and disrupt the one bad copy and potentially correct hypo-PP or hyper-PP. The challenge is you don't want to also snip the bad copy, and they differ by only one. And the specificity of where you're making the cut is this alignment. Each of these lines is aligning matching nucleotide pairs. And this probe is only about 20 nucleotides long, so it's a 1 in 20 difference that we have to develop high specificity, and that's where a lot of the work is. A newer development that's in the past couple of years is through protein engineering, is they took these special scissors and they destroyed the cutting capabilities of this Cas enzyme, and they fused it to another molecular trick, which can edit the nucleotide that will be coded for this, a so-called deaminase contingent G to an A. So now we have not molecular scissors, but editors. So this is really exciting because you would not destroy the mutant copy and be left with only one normal copy, but you would correct the mutant copy and then have two good copies. So these are both strategies that are under active investigation for many human disorders that are completely applicable to periodic paralysis. So while these molecular tools are available, and we're already studying this in the dish, as I'm sure lots of other labs are, I want you to be aware of why we are a long way from just giving you an injection and doing gene editing and helping you. One I've already mentioned is it's absolutely essential, the specificity of where these scissors are acting or where these editing activities are occurring. And that's all about the specificity of this guide RNA probe. And it's going to require a lot of convincing controls in model systems before we're able to apply that. in humans, and this applies to any gene therapy for any human disease. The other challenge is, how do you deliver this to the cells? So gene therapy is happening in some cases where in some cancers that affect blood cells in the bone marrow, because you can take out the patient's cells, do the gene therapy in the dish, and give them back. You can't do that yet for the satellite cells that are the stem cells for skeletal muscle. So there are many groups working on delivery systems to bring gene therapy tools to muscle. So for example, not just in periodic paralysis, but a lot of the muscular dystrophies like Duchenne muscular dystrophy, et cetera. And people are using innovative technologies of using adeno-associated viral vectors to shuttle it in there, or nanotechnology to encapsulate the particles that will be engulfed by muscle. So this is very active. So I want you to be aware that, that these are ongoing, that the horizon is bright. I'm sure this will happen, but there's a lot of work that needs to be done. But this work that is happening is directed to periodic paralysis as well as other inherited diseases. So I'll just end. This is truly my last slide, and then we can either take a gap or go to questions, whichever you want to do. As is traditional in science, I want to thank the sponsors. The National Institutes of Health has funded our lab for 30 years, especially NIAMS, National Institutes for Arthritis, Musculoskeletal, and Skin Diseases. And the Muscular Dystrophy Association has been a real challenge. You've heard it many times. Getting funding to study rare diseases is a challenge. All these sponsors want to help as many people as they can, and it's just a tough choice of where to direct the resources, and we're grateful for everything that comes to us, and I'll promise we'll do everything we can to help you. And these are the names of some of the individuals that you can see on the papers. And I think I'll let Linda now ask the questions. Some of you that came in have more slides, not to overwhelm you. If the discussion does go to the ryanodine receptor that was brought up this morning, I just have two visuals that I think will help clarify if we want to go there in the discussion, but let Linda open it up to the questions. And thank you for your patience in letting me run over.